So thank you, Nelson.
Okay. Give it a moment. It's not recording yet. You should. Okay, now it's recording. Okay, so thanks, Steve. So welcome to everybody. This is our June appointment with the ILS webinars, and it's the last webinar of our first cycle. I'm very happy to be here with Steve Beebe, which is going to be our guest and presenter for this month. Steve works comes from Colombia and works in CGIAR, and have worked there for a long time. Today we are going to talk about breeding in common bean and how this influences our understanding of evolution in common bean. This is also just a gentle reminder that ILS has a conference coming up, our fourth conference coming up next September in Granada, and you're all more than welcome. All the information are on our website, and I will add a small I will repeat the information at the end of our presentation. So without further ado, thanks, Steve. I give you the control, and please, we are all eager to listen to you.
Thank you, Nelson, and thank you, colleagues, for your interest in this topic. I developed this presentation around my personal belief, which has evolved over the past several years, that if you really want to understand the crop, it's very important to understand the ancestor, the primitive ancestor that as it evolved within its native environment. So this is my point of departure for this presentation, which I hope that you will appreciate. Let's see. Nelson, my screen is not responding. Let me see.
It's not responding. Yeah, I see that it's stuck on the first slide. No, it's going.
Okay, doke. So the outline for this talk has 4 points. First of all, the long shadow of the wild ancestor. What traits of the wild ancestor we still see in the cultivated types? Secondly, lessons from the selection of drought tolerance, which I think has been an especially pertinent learning experience. Then I'm going to talk a bit about other legumes from a trial that was done quite a few years ago, which I think still has some very important lessons. And then finally, talk about other species, especially of the tertiary gene pool, which I think have a big potential to contribute to common bean breeding. So visually, this presentation will talk about the evolution of species, how that has created genetic resources with different sorts of physiological strategies with accompanying traits, and how we then carry those traits into a breeding program. Now, you may know that the world's largest collection of genus Phaseolus is housed in CIAT, the International Center of Tropical Agriculture in Cali, Colombia, one of the several centers of the CGIAR international network. Now, within Phaseolus, we have 5 independent cultivated species, but also in the gene bank, we have a number of totally wild species which were never domesticated, which I think have great potential. So here's a quick glance at the wild ancestor of Phaseolus vulgaris. Like many legumes, it's the native seed is very small, dull color, and this wild ancestor stretches from northern Mexico through Central America, down through the Andes and all the way to Argentina. So it's a species which has a remarkably wide latitudinal range in its wild ancestor with some implications, although I won't go into those in depth. Now, when we talk about domestication of common bean, this occurred probably perhaps almost at the extremes of that distribution of the wild ancestor. When we analyzed the wild accessions through AFLPs, we found essentially 4 quite broad groups across that range from Mexico to Argentina. And amongst those 4 groups, 2 of them were domesticated quite amply, that in Middle America to generate the Mesoamerican gene pool, and a second group in the southern Andes, which generated the Andean cultivated bean. And even in Colombia, there was it appears to have been some incipient domestication. But as I said, the 2 major gene pools are that of the Mesoamerican or Middle American gene pool, which we typically associate with small seeded beans, say the small black, small red, or navy beans, and the Andean gene pool of medium to large seeded types, which are often of far more attractive colors and with greater commercial potential. So let's look at a moment at the life cycle of the wild bean. Now, the wild bean exists in what we call a subhumid pre-montane forest. And this is an ecology which is marked by very distinct wet and dry seasons. And what we see here in this diagram is the end of the dry season, when a great part of the landscape has lost its foliage. Most of the landscape is populated by perhaps small bushes or thickets with very few large trees, given the limitation of moisture. And so here we have the wild bean in its natural habitat in the soil. And as soon as the rains return, then the wild bean germinates, as well as the rest of the whole landscape. It comes to life, and the landscape starts to produce a leaf area in response to moisture. Now, the first reaction of the wild bean is to send out a long guide or a long vine. And at this point, the wild bean is looking for some small tree to climb on. And indeed, it finds that tree, and it seeks to climb upwards, because at this point in its life cycle, the wild bean is in a struggle for survival. If it does not reach the top of the canopy very quickly, it will soon be smothered out by the other vegetation. It's a very competitive environment. So at this point, the wild bean is seeking to reach the top of the canopy before it has any sort of entry into a reproductive phase. So if the wild bean were to try to flower and produce seed at this stage, it would simply become extinct, because it would become outcompeted by the other vegetation. So finally, the wild bean gets to the top of the canopy, and now it can multiply its own leaf area, and eventually it will produce flowers and pods, because it now has access to light, which will assure its long-term survival. So, what are the characteristics of this environment, which have created some limitations or defined the character of the cultivated bean? Well, first, as I mentioned, it's an environment which is characterized by shade and limitation of light. So I think that in some respects, even the cultivated bean is very sensitive to cloudy weather. It's also an environment of mid-altitudes, so temperatures are moderate. Typically, this is an environment of perhaps 1,200 to 1,800 meters above sea level. There is abundant rain in season, but as I mentioned before, there are very well-defined wet and dry seasons throughout the year. And with regards to the soil, This is a forest soil, an organic soil, so if it's not an optimally fertile soil, neither does it have severe limitations thanks to the high level of organic matter. So here, even in the environment in which the wild bean evolved, you see certain effects, we can say certain limitations on cultivated bean. For example, cultivated bean is not by nature adapted to the very high temperatures given its origin. So thinking again about this, the development of the Phaseolus vulgaris during its early phase, wild Phaseolus vulgaris emphasizes vegetative vigor for survival in the early phases. So this balance between vegetation and vegetative forces and reproductive forces is very much skewed toward vegetative forces to assure survival of the species. Now, beans are perhaps not unique in this regard, but I think given the environment, beans have a particular tendency toward the vegetative development in the wild state. So one way to define this issue is that of partitioning. In other words, the wild bean partitions its photosynthate and its resources toward the vegetative development. And this issue of partitioning continues to be important for the agronomic performance of common bean in a cultivated state. So partitioning is an issue that I'm going to be talking about extensively through this talk. Now, another way to refer to this that physiologists sometimes use is that of sink strength. In other words, there are certain limitations on sink strength which are inherited from the wild bean. So this is the first point that I wanted to make about how the evolution can impact on the cultivated crop. So let me go on to talking about what we've learned in drought tolerance in common bean. And again, I'm going to be talking about the role of partitioning and what we've learned about partitioning under drought stress. Well, we've published these results in two papers, the first in 2008 and a second in 2012. And so I'll just present a few data extracted from those two publications. This first publication, it was simply a yield trial across environments. And the question that we were trying to answer in this trial was, were we selecting specifically for stress tolerance through our drought work, such that we were creating genotypes which would not respond to favorable environments? I think at least when I started my career, this was the standard dogma that if you select for stress, you will limit your response to favorable environments. So this is the question that we were addressing. First of all, looking at response under drought, and secondly, we planted a set of materials under 3 different environments under good management. And I said that there were probably 60 or 70 genotypes in the entire study. I've simply extracted the data on 3 of these for illustrative purposes. So you can see that under drought, these drought-selected materials had an ample advantage over the commercial Czech variety under severe terminal drought. And we went to these favorable environments. You can see in each environment the Czech responded quite markedly, but we also had materials which were out-yielding the Czech in each of these environments, and frequently quite amply by 700 or 800 kg in some cases. So indeed, we had not limited response to favorable environments through drought selection. On the contrary, we had actually obtained high-yielding materials through drought selection. And in the final column, where it says labeled days to maturity, this was the real surprise, because you can see that some of these high-yielding materials were actually earlier to mature. In other words, we had increased the efficiency of the crop quite dramatically, obtaining higher yield in a slightly shorter growth cycle. So I think this was quite a surprise, which contradicted that standard dogma of 40 or 50 years ago. So in a nutshell, what was happening? Well, we concluded that what we were seeing was improved partitioning from vegetative to reproductive structures. And specifically, we decided that drought stress can reveal differences in photosynthate partitioning very, very effectively. In other words, drought is a limitation on partitioning, but that permits us to separate good partitioners from poor partitioners. Now, we eventually, called this, we settled on a variable that we call pod harvest index. Essentially, this is the concept of harvest index, but in fact, harvest index is, as you know, it would be very difficult to measure on hundreds or perhaps even thousands of genotypes in the field. So what our physiologist Idupulapati Rao suggested was, well, let's take a sample of pods from a row and measure the seed weight as a percent of the entire pod weight, and so we call that pod harvest index. And that has been a very effective parameter for selection in the field. So to make a long story short, the improved partitioning that we recognized through pod harvest index and selected under drought also contributes to yield potential without stress. In other words, through selecting for partitioning under drought, we are touching on some fundamental physiological processes which carry over into favorable environments. Now, the second paper that I'm going to refer to is focuses specifically on pod harvest index as a selection criterion. And I say this was published with a colleague, Teshalia Sefa, in 2012. This is a trial carried out in Ethiopia under irrigated and drought conditions. So the title of the paper, Pod Harvest Index as a Selection Criterion, and the results that can be summed up in this statement extracted from the paper. The correlated gain in drought yield from selection for pod harvest index would be greater than direct yield selection. We'll look at this data in a second, and would also contribute to irrigated yield. So pod harvest index favors drought yield and irrigated yield. Now, the data that I'm going to present is represented here graphically with these photos. I'm going to look at what happens to yield when we select under drought. Well, if we select in drought, what happens in a drought environment? What happens in an irrigated environment? Or if we select under irrigated conditions, what does that do for drought response or to irrigated response? And we're looking at the effect on yield in each of these environments. So again, this is what you see here pictorially is represents the structure of this graph right here. So first we're going to look at what happens if we select in drought and for drought. So here in these data within the red oval, we're selecting in drought, and if we select for grain yield in drought, we get a response of some 56 kg per hectare in drought yield. But if we select under pod harvest index, we can actually obtain 69 kg or slightly better yield response thanks to the pod harvest index, which has a much better heritability than yield per se. So that's selecting in drought for drought. Now, what happens if we select in irrigated conditions and for irrigated conditions? Well, here the response in grain yield is 189 kg. Or if we select pod harvest index and irrigated under irrigation, well, we get 129 kg, somewhat less than direct selection for yield, but still a positive effect. Now, here comes the most interesting part. What happens if we select in drought and we looked at the effect under irrigation? So again, we select for grain yield under drought. The response in irrigation is only 28 kg. That's very modest. But if we select for pod harvest index under drought, we can expect 163 kg response in irrigation. And again, I think when we select for pod harvest index under drought, we're really hitting on those fundamental issues, those fundamental traits of yield associated with pod harvest index. And so this 163 kg is quite substantial. And it isn't even far off of selecting for grain yield under irrigation. So I think it emerged as a very interesting study result, either through those conventional yield trials that I presented before, or here quantified within a given population. So time and again, I come back to this issue of partitioning, that drought stress reveals those differences that we want to select on for photosynthetic partitioning. So in drought, we can identify the best partitioners with more confidence because drought expands the genetic variability in the field and increases heritability. Now, this is the concept of harvest index, but pod harvest index is much easier to implement on large numbers in the field, simply because pod harvest index would imply gathering huge piles of biomass and taking it to laboratory and drying, threshing. Pod harvest index is logistically much simpler, and pod harvest index, therefore pod harvest index or PHI is much simpler to do than harvest index, and still very effective. And recognize that I think the same physiological principles. So to wrap up, we're going to look a bit more extensively at an integrated model, which we conceptually developed based on this experience. Now, here I'm talking about bush beans as opposed to climbers, and I'm hesitant to extend these principles to climbers because I think it's a very different physiological animal. But if you look at bush beans, typically between planting and physiological maturity, flowering is pretty much in the midpoint of that growth cycle. So what we thought of doing as an idiotype to improve yield would be to expand the vegetative phase. In other words, we would like to get a crop with perhaps with more biomass, perhaps a more rustic root system, still maintaining very good leaf area. So we would like to increase the biomass by a bit later flowering. But when we get to flowering, we want to have a decisive shift to the reproductive phase. We don't want a crop that's going to be stuck in vegetative development. Having developed a larger plant, we want that plant to put its energy into reproductive processes. And then, of course, we want to have good grain fill, and eventually we want to have a quick dry down of the crop, because I think for farmers are really not interested in physiological maturity. I think scientists focus on physiological maturity, but farmers want to have a crop that they can harvest, and so that's where the quick dry down comes. So this was our model, I think, for improving yield, perhaps under stress, and also probably yield potential. Now, it has not been all that easy to obtain this, and I think that the difficulties in meeting this model, I think, have a lot of lessons as well. And especially as what I represent here, what is difficult about this model is that decisive shift to the reproductive phase. I think it's almost an intellectual construct which is very difficult to meet biologically, because in reality, flowering does not represent the whole shift to the reproduction. Actually, the transition to reproduction is a long process. So it's perhaps over at least 2 or 3 weeks. I mean, it starts several days before flowering with flower bud formation. It reaches a peak in flowering, but even after flowering, the crop still needs to be moved more decisively toward its investment or its partitioning to reproductive development. For example, you can still have embryo abortion after flowering because the crop is not, again, investing sufficiently in reproduction. So this phase, this transition phase, I think really requires intensive study to understand and to break perhaps a yield barrier. So here I'm turning the clock back 40 years to what for me is, I think, is one of the most illustrative and informative trials ever done in legumes. Our physiologist at that time, Douglas Lang, who passed away a couple of years ago, he did perhaps the simplest trial in the world. He obtained seed of as many of the tropical grain legume species as he could, and he did a yield trial. And here he has 3 species of Phaseolus, Acutifolius, Vulgaris lunatus. He has several species of Vigna. He has Radiata, Angularis, Caupi, Unguiculatus, and he has soybeans, and he has pigeon peas, and he also has ground nuts, Arachis. Now the Arachis didn't do well in this environment because he was working on a very heavy clay soil. And I think the Arachis are not able to penetrate that soil for seed production. But essentially, he did a yield trial and measured yield of these multiple different species, and then he mapped that across a variable that he called leaf area duration. Essentially, this is looking at how much leaf area exists over the cycle of the crop, and this is kind of like in disease rating, you talk about the disease progress and area under the disease curve. This is like area under the leaf area curve. So in results, he comes up with a very nice curve, which would suggest that yield is a function of the photosynthetic structures. And I suppose that doesn't sound very, very dramatic. I think that almost very much expected, but I think demonstrated quite dramatically here. So what's unusual about this data? Well, this is this is the curve you get when you look at yield in terms of tons per hectare. But what if we think about energy yield instead of simply grain yield? Because the soy and the ground nuts have much greater energy in their kilo per kilo due to their high protein and oil content. So the soybean has about 40% more energy per kilo than the other legumes, and ground nuts, given it's almost entirely protein and oil, they have 60% more energy kilo per kilo. So if you ask, okay, what would an energy curve look like? Then these 2 species are just way off the curve. Even ground nuts with poor yield, would be above the curve as tracked here. So I think that so this has some, I think, some very important implications or about how we interpret, what's happening with photosynthesis. And here I say that the photosynthesis is capable of much more than what we see in Phaseolus or Vigna or Cajanus. Somehow these other species, assuming that the photosynthetic capacity per leaf area is comparable, these other species are simply not making good use of their photosynthetic capacity. So in some sense, these other species are lazy in creating yield, or at least in creating energy. So why is this? So how can soybean and groundnuts produce so much energy? I mean, in final analysis, energy is photosynthesis. Now, is their photosynthesis more efficient? I mean, they are polyploids. Is that contributing to efficiency? Well, I have a different hypothesis, and that is that the capacity to store more energy minimizes the feedback mechanism to photosynthesis, and therefore it keeps photosynthesis much more active. So these two species can store energy as oil, as protein, and that permits them to create more energy. So is the capacity to download photosynthate from phloem to seed, or say the sink strength, is that capacity to download photosynthate limiting on yield? In which case, don't we need to create more sinks capable of absorbing photosynthate in these other legumes? Well, I assume that there are breeders in the audience, and the breeders might be saying, well, you silly fool, I mean, that's obvious. We need more pods and more seeds. But again, let's go back and really think what's happening here and see what the barrier to getting more pods and seeds on the crop might be. So let's go back to Doug Lang's curve again and say, well, if that curve doesn't reflect energy production, what does it reflect? And I think the answer might be that it reflects the physical size of sink as a function of photosynthesis. And that, in other words, at a certain point when those pods and seeds are being formed, that is when photosynthesis is limiting. So the photosynthate availability at the moment of flowering determines the number of pods and the number of seeds. So that moment of pod formation is the critical issue in determining yield. Again, probably not a radical conclusion, pretty logical by any standards, but I think we can maybe modify our thinking about this again think in terms of partitioning, and suggest that partitioning of photosynthate is limiting at flowering much less than at grain filling. Because think about this, the photosynthetic structure or leaf area doesn't really change greatly after flowering for these bush beans. They have pretty much the same leaf area, the same photosynthetic capacity, but again, we're not making use of that at that critical moment of flowering when we need it. So somehow the key to improving yield is salvaging more reproductive structures during this transition phase, and so we need better partitioning to the reproductive phase during that transition. We need to speed up somehow that investment in flowering because we have the photosynthetic capacity, but it's not being used, it's not being directed toward the reproduction. So we need to salvage those flowers and those embryos and maintain them until the crop is more fully dedicated to its reproductive partitioning. So can we salvage more flowers to set pods? Can we salvage more embryos? Again, how can we make the crop partition at that critical moment? So one option would be to improve, say, the number of seeds per pod. Say if in a typical pod we have 8 or 9 ovules, we're only taking advantage of perhaps 4 of those. We would like to salvage more embryos and have perhaps in this case 7 seed instead of 4 seeds. So that all sounds fine and good, but if you're a legume breeder, you're probably asking, well, how are we going to do this? Where do we get, say, the genetic variability to modify the partitioning around flowering or pre-flowering? Like this is where we start getting interested in other Phaseolus species. Now, the genus Phaseolus originated or evolved over a remarkably wide agroecological range. You have the vulgaris in a subhumid environment. You have the secondary gene pool, its closest relatives, in frequently a totally humid environment. And the species most common or most widely known in this of these species would be Phaseolus coccineus, or the scarlet runner bean. Now, what I mentioned about the wild Phaseolus vulgaris applies much more intensely to coccineus and other species, such as that the vegetative vigor of the wild coccineus needs to be very, very vigorous to survive. It's in an even more competitive environment, so the coccineus likewise needs vegetative vigor to survive in the wild state. And so as a species, coccineus tends to have a low harvest index. As I say, it's kind of an exaggerated case of what I described for common bean. Now, if we go to the other extreme of the agroecology, we move into a semi-arid or an arid environment, this is where we find the tertiary gene pool. And here the species par excellence is Phaseolus acutefolius, or tertiary bean, the most common, most widely recognized or widely known species. And here, the tertiary bean doesn't depend upon vegetative vigor for survival. On the contrary, in the wild, it has to produce seed as fast as possible to survive. Its survival depends on rapid seed production. And you think about that physiologically, that implies probably better sink strength and high harvest index. So again, we look at these survival mechanisms under these very different contrasting environments. Physiologically, these have evolved very different mechanisms for their respective survivals. And so that makes the tertiary gene pool very interesting as a source of agronomic traits when we think in terms of sink strength and harvest index. So here at a glance is that environment where the tertiary gene pool evolved. Our germplasm expert, Daniel Debuc, would suggest that acutefolius is probably at the lowlands where the water accumulates down there in the valley, whereas other species like montanus or filiformis are up here on the higher ground where water never accumulates and where the droughts and drought stress is truly intense. Now, but acutefolius in any case in our drought trials is remarkably resilient. On the right-hand side of the photo, you see a drought-tolerant vulgaris, and under this stress of about 100 millimeters and high temperatures, the vulgaris is practically dying, while the acutefolius is developed vegetatively. It's at pods, and within pods, the seed is well filled. Tertiary is typically very small seeded, but in any case, You can see that here we have 7 seed per pod under these very, very harsh conditions. So again, the tertiary gene pool is really remarkable in terms of the traits that it offers to the breeding of common bean. Now, for years and years, people did attempt to use tertiary bean for breeding. Originally, it was used as a source of bacterial resistance, but in any case, it required embryo rescue in the laboratory. This implied taking very small pods off of the mother plant, which is typically vulgaris, taking those very small pods, excising the embryos, carrying them into a culture medium in the laboratory with small plants that germinated, carrying these into a solid medium and eventually into soil. So, and it was a very laborious process, very difficult to replicate and imitate say, a natural process of seed development, and extremely limiting in terms of, say, of a mainstream breeding program. Was simply not by any means close to the scale of a breeding program. Now, the surprise, and this is where, one of my breeder colleagues says that breeding is really a lot of dumb luck, and this is a case of, I think, of dumb luck. One of our assistants was playing around with some older breeding lines, some old interspecific breeding lines, and he was pollinating with Acutefolius. So those breeding lines were already interspecifics, which had some degree of integration from Acutefolius through this very laborious laboratory process. But when he pollinated some of the lines with Acutefolius, he found that they were capable of developing a seed on the plant. So essentially, he had discovered some bridging genotypes which were cross-fertile between vulgaris and Acutefolius, and which don't require the need for embryo rescue, which was a remarkable and highly relevant discovery. And so this has really broken the logjam on using the tertiary and other species of the tertiary gene pool. So we have used these genotypes quite widely in the program. Obviously, we start looking for heat and drought tolerance as one of our primary objectives. But we've also found that they contribute to higher seed iron in our biofortification project to raise the iron concentration of the grain. I'll talk about that in a second. We also find insect resistance, actually several insects. Again, the evolution in a dry environment seems to have favored that. Yes, we have potential for disease resistance, and I'm really convinced that we also have the potential for, I think, for higher yield, thanks to the potential for better partitioning to grain in this, coming from this dry land evolution. So I'll give 2 examples of progress in this regard. I think here I'm talking about interspecific crosses with the Andean gene pool. If you're familiar with bean breeding, you might know that it's been especially difficult to increase yield and stress tolerance in these large-seeded Andean types. Here in the photo in the center of the screen, you see pods from Andean types with 1 or 2 seed per pod. Other embryos have aborted. So in drought-sensitive materials produce only 1 or 2 seeds. Interspecific lines can produce 4 or 5 seeds per pod, and with a great reduction in abortion, and still have a very well-structured and well-synchronized genotype with good dry down at the end of the cropping season. So I think even in the Andean types, which have been so difficult to improve, we're already seeing some benefits coming from interspecific crosses. Now, I mentioned that we're also getting some results of high iron coming from these species. Within the graph, you see the lines identified with red font. DOR 500, Cal 96, scarce in this trial, not even with 50 parts per million iron within the grain. One of our best vulgaris-derived lines, SMC 33, has 78 ppm, and with lines derived from, let's say, parvifolius, we can get up to 90 or 100 parts per million. Well, why does this happen? Well, again, we go back to evolution, because parvifolius originated not only in a hot, dry environment, but in an alkaline soil. And alkaline soils have very low iron availability. Therefore, parvifolius has its genes calibrated to take up more iron from the soil, and we can exploit that, say, for biofortification with high iron. Parvifolius is iron hungry and is receptive to absorbing more iron. So here we have a case related less to climate per se, but to soil. I think we seldom think about soil adaptation, but here we have an example where evolution also is expressed with soil adaptation. So over the past few years, we had a project to tap high temperature tolerance from the tertiary gene pool. We did GBS on about 300 lines, and out of those 300 lines, we identified about 14 with introgression, and which summed across the entire genome. The introgression of these lines represents about, we estimate, 59% of the acutifolius or parvifolius genome. So, and this is, to me, this is quite a remarkable achievement, which bodes very well for tapping even more traits within this gene pool. And I think that these lines might well serve as additional bridging genotypes to facilitate further introgression, given that we expect to have, say, better chromosome pairing and more chiasmata for introgression. So I think that wraps up what I wanted to share with you today. Thank you very much for your attention. I guess we have a few minutes for questions if anyone has anything on their mind.
Thanks, Steve. It was a very interesting presentation of yours. And yes, we have usually we have a few minutes for questions. So if anybody from the audience want to intervene, now it's time. You can either unmute yourself and talk, okay, or Tom, please, you have the mic.
Yeah, thank you so much, Steve, for that really nice talk. I wonder if you could just clarify a little bit about your pod harvest index, just sort of the methodology that you used. Were you simply harvesting pods and then weighing the seeds versus the weight of the entire pod? So is it a function of having thinner pod walls?
Yeah, we haven't looked at in detail about, say, the pod wall thickness. I suspect that that might figure in the equation, but yes, it's essentially its process is very much what you described. We go to the field at say at maturity. We take a sample of pods. We standardize within the mid part of the canopy. within a row of maybe a 5-meter row, you would take 20 pods, dry them down well, then weigh the entire pods, thresh, and weigh the seed, and that generates our pod harvest index.
Okay, thanks a lot.
You're welcome.
Anyone else? Yes, who's Paolo?
Hi, Steve. Thank you also for the remarkable clarity of your talk. I have two questions, actually. The first is still about the pod harvest index. Everything clear. The only thing a little bit unexpected to me was the fact that apparently so selecting for a high pod harvest index under favorable conditions.
Acho que fomos só nós, porque o resto está cá tudo.
You got even
Já voltei.
a negative progress under drought. If I understood well your table. And the second question, I was wondering, I mean, you provide really a very interesting example of the usefulness of a tertiary gene pool. Was there any, I mean, clear unfavorable traits, any clear genetic load, I don't know, for other quality traits? Because in the end, domestication was a long process on common bean. So I wonder if there were also manifest disadvantages in this crosses, no? Wide hybridization crosses.
Well, let me zoom back to the okay. Here, see, okay, now you picked that up. The selection under we select under irrigation. The response in drought could be negative. I don't know. I don't have an explanation for that. You caught you're quite attentive to that, but no, I don't have an explanation for that. You asked if there are any unfavorable traits which come through. I think you're referring to the use of the tertiary gene pool. Surprisingly, I have not found those traits. I would expect the negative traits in terms of seed size, but that seems to be quite relatively easy to overcome. And frankly, and here I'm going to stick my neck out, but when we look at the structure of the pedigrees, it appears that we are working with some major gene effects, which make it relatively easier to deploy and to introgress. Now, one kind of initial kind of unfavorable tendency is that when I talk about this model of lengthening out the vegetative phase and then getting a decisive shift to the reproductive phase, when we put a lot of emphasis on that decisive shift, we can get even a shorter vegetative phase, and therefore an earlier crop and a bit lower yielding in terms of the biomass. But I think we're starting to see some genotypes which kind of break that tendency. So I'm confident that we can overcome even that tendency. But in essence, you know, I've been surprised that it has been so easy to introgress from the tertiary gene pool. I would not have expected it, but I think that essentially we're kind of extending that natural domestication syndrome, where we take if we take the wild vulgaris and we try to create a crop with better harvest index, better partitioning, and when we go to the tertiary gene pool, kind of extending that tendency of the domestication syndrome. toward better harvest index. So, but say negative impact in terms of say plant type, which could occur, but it doesn't. Small seed has been easy to overcome. So I've been very surprised, frankly, that we have not had more problems of negative traits.
Thanks.
Thanks. Any other questions? So I have a question for you, Steve, since, okay, so I'll wait. My question will wait after Juan Osorno, please.
Hi, Steve.
Yeah.
I agree with you. I see the same issue of, during flowering, you get a lot of abortions, right? I'm always scratching my head thinking how we can make sure that that flower that was produced by the plant is going to be set into a pod, right? And even in good conditions, I mean, it's pretty obvious that during in stress conditions, there's going to be some flower abortion and stuff like that. But even under really good conditions, optimal conditions, I still see that the plant somehow decides I'm not going to set all these flowers, and some of them are dropped, right? And the question is how we can make sure that those flowers will set into pods. Where is the key for that? I mean, it's pollen germination. What is that physiological trait that we need to really keep track of to make sure that we're going to make sure that we're not wasting energy in the plant by doing all this, right?
Well, that's a real essential question. And I mean, I'm talking about partitioning. I'm kind of begging that I'm avoiding the hard question of what is really going on.
Yeah.
Because I don't have an answer. I think that yes, I can say we need to get more photosynthate into the reproductive structures, but like what is the primary effect? Is it salvaging embryos? Is it better, say, pollen tube growth? I have no idea. Yeah.
Yeah, that's $1 million question. I agree. Yeah.
Yep.
Thank you.
Yo.
Okay. Other questions? So, Steve, I'm curious, your presentation has been mainly about breeding, but given that for the harvest index, you said that it had a very high heritability. I wondered if that has been investigated from the genetic point of view. I guess it would be a good candidate for association studies and the similar.
Yeah. Well, I think there are a few studies looking at QTLs for pod harvest index. I think one that I believe the one that was especially, I say, successful was at one at UC Davis, Paul Gepps and a student, Jorge Bernay out there. I think they found a QTL for even 17% of the genetic variability.
Mm-hmm.
So that's pretty dramatic, but for a trait which has shown consistently high heritability, I've been a little bit surprised that we don't find more really significant QTLs. But
Sure.
I think this is kind of similar to Juan's question about what's really happening, and no, what we know so far is pretty limited.
Okay. I don't see any more raising hands. So I'm going to wrap it up. Thanks again, Stephen. It was a wonderful, very interesting presentation. To everybody else, I thank you, all the people present, for this will be our last appointment for this first cycle of webinars. I hope to see you all in Granada this September at our 4th ILS conference. And for everything else, thanks and goodbye.
Okay. So thank you, everyone.
Goodbye. Thank you, Steve.
Bye.
Bye, everyone.
Bye.